So welcome everyone to the above and below ground linkages of forests and soils. We have some great presentations up ahead of us, and so and we have one missing speaker. So Chris Miller was unable to attend. He is ill, and so I'm not going to be too hard on my speakers with time, but I'll try and keep them to about 15 minutes for sure. So our first speaker today is Rob Fleming, and Rob is a forest ecologist with the Canadian Forest Service at the Great Lakes Forestry Centre in Sault Ste. Marie, Ontario. He has worked for quite a while on harvest-related impacts on biophysical soil processes and regeneration, silviculture, and stand dynamics in boreal and northern temperate ecosystems. And I'm just going to let everybody talk about their title and all of that. So Rob, take it away.
Thank you. Okay, so yeah, I'd like to start by acknowledging that we are on the traditional territory of the Huron Treaty, Robinson-Huron Treaty, which includes the Batchewana and Garden River First Nations, and that includes both Sault Ste. Marie, where we are virtual, and the tolerant hardwood ecosystem at Turkey Lakes, which we'll be talking about. So, tolerant hardwood forests, microclimate. This is my thing. So I will apologize right offhand if you hear more about microclimate than you do about soil processes. I think Dave can follow up on that in the next presentation. On the bottom left, I'd like to acknowledge right off the bat, Deb Massa. She has done much of the microclimate instrumentation and work. She's retired, but she was key to having this this study going forward. And on the top right, a person probably many of you know, Paul Hazlitt, and he has contributed much of the work in terms of soil nutrition. Well, in terms of location, and we did try and locate a map on the top left there that included all of the three countries in North America. So we're talking about the range of sugar maple there, and if you look at Lake Superior in the middle there, the largest lake, that's where our study is located. On the top right, it's a zooming in there, and this is called the Algoma Forest section that is highlighted in the light green, and we are just slightly below Wawa. And on the bottom right, a tolerant hardwood forest. We call this an older forest. It's not a old growth forest because these forests in many regions, or in many locations in this region, have been previously high graded for sugar maple, but particularly for yellow birch. So one of the silvicultural prescriptions here, many of them actually revolve around increasing that component of yellow birch and other mid-tolerant species. So if we're going to talk about microclimate, and I'm always about talking about soil temperature and moisture and all that, I actually realized when I was putting this slide together, we have to talk about regional climate first. And in terms of regional climate, we have 4 distinct seasons in this area: a winter, spring, summer, and fall. And by the way, that fall picture, that didn't come out as some postcard. That is actually a Turkey Lake. So just so you know, and you can the ruggedness of this, the hills that emerge, and we have distinct east-west face or east-west running ridges, so we have very distinct north and south aspects. And getting back to winter, we have about 5 months of winter here, and with large accumulations of snow, largely lake effect snow that comes off of Lake Superior, similar to some of the amounts that might be received in northern Michigan or areas, northern Michigan areas on the lee of other, say, Lake Michigan. And as you can see, Deb doesn't always get to pose in a nice setting in front of a climate station. So in terms of the study itself, we're looking at 3 particular factors that were that have silvicultural implications: the degree of canopy opening, soil disturbance, and here in these systems, The soil disturbance is largely logging disturbance, either logging or skid row disturbance. And also we're looking at that effective aspect. The measurements we'll be talking about here that contribute to this study, microclimate, particularly I'm going to look at understory temperature and moisture. We have mensuration in terms of woody species greater than 2 centimeters in diameter, just to give you a sense of how these forests in terms of productivity, of overstory productivity are developing. Some solute concentration work, and so there we're looking at how, what is the concentration of solutes near the bottom of the rooting zone? How are these treatments having an effect on that, and potentially then on nutrient leaching? Also soil respiration, a lot of the work that Kara Webster has done, we've tried to incorporate some of that here, and mineralization. And finally, we'll also talk about LAI and understory light, because that drives a lot of this stuff. So this is some of the instrumentation. On the top left, you'll see the chamber used by Kara and her colleagues for measuring soil respiration. Just to the left of those are some lysimeters Paul was using to collect that solute concentration. In the bottom left, one of our microclimate stations here, it's underneath the forest canopy. But if you look, you'll see there's actually a pyranometer sticking off to the top left there, measure understory light, which we'll come back to. Some other types of standard climate station temperature, humidity measurements. And then on the right is one of our soil disturbance plots, where we're also looking at, those are tensiometers there. There's also TDRs, some soil temperature sensors. It's kind of hard to show those. The boardwalk, so we're not trampling the site. And then in the top right, for you soil aficionados, this is a soil pit, one of our typical sites with sandy loam soils. They're fairly rich. They're 60 to 70 centimeters deep, ablation till over a compact basal till. And if you had seen some of the earlier presentations that with regard to Turkey Lakes and watershed studies, then that basal till ablation till interface is really important. Okay, so when we impose these treatments, let's go back. We can see in terms of basal area, we removed about 80 to 90% of the basal area in the what we call the clear cut treatment or the open treatment. We removed about 40% to 50% of the basal, maybe 40 to 50% of the basal area in the partial canopy. Those were termed selection and shelterwood. If you're reading other publications on Turkey Lakes, that's how they'll be referred to. The truth is, when you're looking at terrestrial processes on a plot level, those two treatments didn't vary much at all. So we've combined them together. And we're also showing here the effect of the north versus the south, pardon me, the effect of the soil disturbance on those various treatments. So initially and through year 7, we're not seeing a big effect in terms of the overstory basal area. Now we are seeing an effect on the understory, but that's not really contributing to the woody biomass. However, by year 20, we're getting close to, but not up to, the same levels as in the unharvested forest. So the takeaway here is that it's taking a long time to build up the basal area, or relatively long, in these ecosystems. So, and that's to be expected. That's how these forests would develop. So if we look at LAI though, and keep in mind that the leaf area development occurs much more quickly than the woody biomass development. And when we start looking at that in terms of, on the left, we have canopy and aspect. We can see that at year 1 and through year 2, it's fairly in the partial and the open or clear-cut areas, we have a reduced leaf area. But by year 5, it's really building up. And by year 9, we have almost the same amount of leaf area, overstory leaf area, in the treatments, in the partial cut and the open or the clear cut, as we do in the uncut forest. And if we look at the effect of disturbance on that, we see kind of a similar situation whereby by year 9, we don't have a lot of differences between the different levels of opening and disturbance. So this is what it all comes down to. This is a slide showing in the darker brown and the mid brown, it's the difference in relative shortwave radiation, that is the amount received in the understory or at the forest floor compared to what occurs in the open. And the point here is to look at really at if you look at the very bottom of the slide, we've divided this into 4 different seasons. And those then have reflect this amount of light that's received in the understory. So during the summer period, in the middle of the diagram, you've got very little light, which is really, you could call that energy, very little energy received at the forest floor in this uncut mature forest. However, during the during the leaf off period, so to speak, you have a substantially more radiation received at the forest floor. Now, this is, I guess that's obvious, but at the same time, it's really what drives everything here. And the reason, by the way, that we're not getting full light on the forest floor during the non-leaf on period is because we still have a lot of interception occurring by the boles and the woody branches. And that's particularly important when you have low sun angle, which reflects what we've got on the right-hand side of the diagram in the fall, in November and December. Superimposed on this, then we put what the soil temperature might be, or what the average soil temperatures were, and both in the clear cut and in the forest. And as you can see, when we have that leaf off period, we don't have a large difference between the soil temperature in those two conditions, either on the spring or the summer. Where the real differences occur then are during the growing season, when we have the full canopy in the mature forest and not in the open. So looking again at the diagram, we have winter, which is about 5 months. We have spring, which is about 1.5 months. We have the summer, which is about 3.5 months, and we have fall, which is about 2.5 months. So whereas we say there's 4 seasons, these seasons are distinctly different in their length. So we showed you the leaf area index a little earlier, but there's a difference between leaf area index and the amount of light actually received by the understory. And that's what this diagram shows. The diff-N is actually diffuse non-interceptance. It's measured with an LAI 2000 instrument. But the point is that very early on in the development of this study, we start to see few differences between treatments. And by year 5, we've actually seen, and particularly by year 9, but certainly by year 5, everything is converging. So in terms of the amount of energy received at the forest floor, where these soil processes are going to take place, and which are largely driven by temperature, that we can't expect to see, or we might not expect to see, big differences continuing to occur. And so if we look at that on a seasonal basis, this is immediately or very close to, soon after harvest. We can see here we're looking at canopy opening and disturbance, and the FF is includes a forest floor, so not disturbed. The NFF is no forest floor or disturbed. And you can see that the forest floor disturbance, say if you're looking for open, the red has a much bigger effect, has a substantial effect. And indeed, the ones for the open and the partial cut have higher temperatures generally earlier in the season than do those for either of those treatments when the forest floor is undisturbed. In terms of soil temperature then, we have on the left hand, it's the importance of disturbance, and you can see that over these periods of time, over the first 10 years, things converge, no big difference. We are still seeing a difference though in terms of north versus south aspect. So aspect has an enduring effect. And how are we doing for time, Deb?
You need to wrap it up.
Okay. we will go forward here. Organic matter in the, this is the organic carbon in the solute concentrations. It increases, but largely this increase occurs between 6 and 8 years, reflecting the degradation of larger woody material. If we look at NO3, on the other hand, rapid release of NO3, but later in the period of in the establishment period, we see it actually for the open canopy, for the clear cut, falls below that for the other treatments, and we think this represents largely the uptake by the developing understory. So I will go to quickly soil respiration driven by soil temperature. Well, there's a surprise. And so if we model that, we find that there is greater soil respiration with the canopy opening, with the clear cut, and also on the north versus south aspect. We might say, though, that when we have disturbed soils, we get less. They have similar sort of temperature functions, but the soil respiration decreases by about 35% when you have disturbed the soil. So there's our picture again. In this rugged northern tolerant hardwood ecosystem, we have rapid microclimate and soil process recovery. But we haven't looked here at the longer term effects of species composition and stand dynamics. And if we have more intolerant species, semi-tolerant species like yellow birch coming in, then that may indeed have an effect on the longer term soil processes. Seasonality, we talked about 3 to 4 distinct seasons, but of different lengths. Our microclimate processes, certainly related to light regime, related to soil disturbance. They do affect soil temperature largely, a few effects on moisture, and those soil disturbance effects, forest floor processes, but species ingress as well, and that's not something we've touched on, but yellow birch and pin cherry are coming in, and they may have distinct effects on nutrient cycling through their own litter. Finally, aspect, that's important too. And so I'd like to end by saying that we share these forests with all of our relations.
All right, so I'm going to sort of continue what's been sort of a common thread or theme throughout actually this the workshop, and we had a special session on it yesterday as well, and it's dealing with this idea about, are we emulating natural disturbance, with a big question mark, and we've had a lot of talks about that, and what's the control, and how do we actually track that. For this talk, it's going to be from a soils eye view. I'm going to look below ground. Specifically, we're going to be looking at the temporal patterns following disturbance, following wildfire, harvest, and salvage, and look what's happening with soil carbon and soil nutrient pools. I thought, since we're even though it's virtually, we're having this meeting in Sault Ste. Marie or in Ontario, I thought I'd just give you 2 really quick sort of forestry facts. The area, the outline here is what we used to call the area of the undertaking, but basically this is the managed forest landscape of Ontario. This is where we conduct forest management in Ontario. It's about 37 million hectares, so a pretty big piece of real estate. And I think Lisa had pointed out in a couple of times that largely Crown land, a little bit more private land in the southern portion of it, but certainly across the Boreal, it's pretty much all Crown land. And in the other inset there, just looking at the total productive forest area, again, most of it, over 50% of it, is still in mature or over mature forest conditions. So we're still, to a large extent, still cutting primal forests, at least across the Boreal. In terms of harvest amounts, historically we were over 20 million cubic meters per year. That we like, I think a lot of places, we had a downturn in the forest industry starting around 2006 to 2009 with a number of mill closures, especially pulp and paper closures. But it has rebounded a little bit. So we're about 14 to 15 million cubic meters per year. So it's a big deal to Ontario's economy, for sure, as the forest industry. I just wanted to put this one up. I think like other jurisdictions, it amazes me now that it's been almost 30 years, but a real shift in our thinking from timber management, where we're really supplying, timber for the forest industry, to the sustainable forest management. And there are a number of forest policy drivers. And so I've got a few of those reports there, but a big one was our forest class environmental assessment on how we do forest management on Crown lands. It was a multi-year process, and that really set the stage for this. The Crown Forest Sustainability Act of 1994, that replaced our Crown Timber Act of 1951. So again, going from supplying timber to, again, this concept of a broader ecosystem management and ecosystem services. So built right within the Crown Forest Sustainability Act are these things like, conserving biological diversity, maintaining ecological processes, and this interesting word that we've had lots of discussion around, emulating natural disturbance and landscape patterns. But I'm sure it was Ellen, but it may have been others, and I start this off in a lot of talks. Wildfire and harvest are not the same. So why would you expect them to be the same? Again, one is the wildfire is a pyrochemical process, right? That in many cases is a stand replacing fire. That's what you see here on the left hand side. But it kills, but leaves all that standing dead material that eventually falls to the forest floor. But it does consume a portion of the forest floor. And if you're interested in soils, that old horizon is a big carbon and nutrient pool in a lot of these mature boreal forests. In the center plate here, we've got the clear-cut harvest. That again, it's a mechanical disturbance. They come and physically remove all the overstory trees, but the forest floor is actually largely left intact. In many cases, there's mechanical mixing, and certainly if you do site preparation, but it's all retained on the site. It just a lot of times it's maybe translocated a bit through the profile. And then over on the right-hand side, we have salvage logging, which is really a double disturbance, right? You have the pyrochemical impacts of the wildfire, but then you come in and do the mechanical removal as well. So here, what we really want to sort of look at these three types of key disturbances and how they're affecting soils. I think Terry had it in our session, had these two. Again, the idea is that these are stand replacing fires, but then we have these stages of stand development, right? We go through a regeneration phase, crown closure, we go through the stem exclusion or self-thinning phase. And certainly above ground, we can see those changes, and many people have recorded those over time, the rebuilding of that forest floor, that continuous feather moss layer. But what's going on below ground with these? And then of course, if you look at post-harvest, early on, they do not look the same, right? You have these a lot of times planted in straight rows in the regeneration phase. But as time progresses, you start to look at those forests, and it becomes harder and harder to tell the difference between, the harvest or the wildfire. But again, what has happened below ground is of interest. So that leads us to three questions. So the first one is really, what's the temporal pattern, the loss and recovery of soil carbon and nutrient pools following wildfire? So we would use that as the natural disturbance benchmark, right? The temporal pattern, though, not at any one point in time. And then the second one, of course, how do the post-harvest versus the post-wildfire temporal patterns compare? And again, here, we would anticipate there should be some differences initially because they're the different types of disturbances, but do we see convergence or do we see divergence? I think that's a really important question. And then when we get to the salvage logging, which is that double disturbance, do we see greater declines and slower recovery in those soil pools? So for this study, what we did, again, this was a chronosequence approach, right? The idea of replacing time, repeat measures at one particular location with space. So multiple sites. And in this case, we have 84 sites. This is sort of a labor of love, I guess, for me. I started doing some of the sampling in 2002 and started building up more sites with different studies and generally variations on a theme in terms of how we did the sampling. But we do end up with, we have 84 sites, 37 fire sites and 37 harvest sites, far fewer salvage sites. So we don't do a lot of wildfire salvage here in Ontario. It's very opportunistic. You pretty much have to have a road right next to it, because you're not going to build a road to go in and get this sort of lower value salvage wood. And you generally have to get in there within 2 years, because the wood boring insects get into the wood pretty quickly. Oh, the other thing I guess I should point out is that the numbers in the brackets under each of these, those are the actual sampling points. So we do have multiple sampling points within a stand. So we do try to represent the within stand variability as well as the between stand variability. Here's a really crappy map, but down here, you can't really see it. This is Lake Superior, Lake Gitchagumi. This is Thunder Bay, Lake, oh, I can probably go back. I won't bother going back for that one. The idea with those points, I wanted to just sort of point out, there's a pretty good interspersion, at least with the harvest and the fire. We were able to kind of intersperse them. They're all in ecoregion 3W, which is the Lake Nipigon region. Rainfall, annual rainfall is around 750 millimeters, and mean annual temperature is 1 degree Celsius. The other important thing with when you do a chronosequence, right, is you try to make sure that the stand, the site, the soils are very, very similar. It's never easy to do across, you know, when you're trying to pick other stands, but for these, they were all jack pine dominated sites. They're all 80% to 100% jack pine. The soils were glacial fluvial deposits, outwash sandy sites, and generally well-developed cumuliferic podzels. The other thing I'll just point out here, when you look at the profile, you can see where the sort of functional rooting zone is, where a lot of the fine roots hang out in that upper 20-25 centimeters, and that's the focus of what most of our sampling has been, is in the upper 20 centimeters plus that LFH layer, that O horizon. So when I talk about total soil pools, that's what I'm talking about, is that 20 centimeters plus LFH. Okay, so into the results. Okay, so to our first question, so what happens after wildfire in terms of soil pools? And you can see carbon and nitrogen. I don't know if you can see my little arrow moving around there, but just from the fire itself, the consumption of that forest floor, we see a decline of about 20 tons of soil carbon, or like I say, largely the loss of that deep O horizon. And then you see just kind of a long, gradual return back to the pre-harvest levels. And that's largely that, again, there's that coarse wood, that standing material that then falls down, slowly decays, sloughs off its barks, and that material slowly becomes incorporated into the forest floor. And then, of course, once the stand is developed, you start to get the self-thinning aspect of it as well. Phosphorus, a little bit different. Phosphorus, we always see post-fire this little pulse of phosphorus, and this is Bray's P, so it's an estimate of available phosphorus. But then you have this very long extended decline, and actually you'll probably lose about 60% of the total pool of phosphorus pool out to about 40 years. But then when self-thinning kicks in, it actually rebounds fairly quickly and stabilizes, you know, at about 60 odd years. And calcium largely doesn't seem to care. It just sort of stays right there at around 500, a little blip up, a little blip down, but it pretty much stays right there. Okay, so for the next one, so what about harvesting? What happens there? So if we just look, and actually carbon, nitrogen, and calcium largely have the same pattern. And guess what? The pools are actually higher. Again, we retain that old horizon. We redistributed it a little bit, but so it's pretty flat. And then you do have a lot of logging debris. And even though we did do full tree logging, we still find 15 to 25 tons of slash is left after an operational full tree harvest on these types of sites. Broken branches, tops, those sorts of things. So we have this increase to about 40 years, and then we start to see this convergence. It actually starts to go down, and there isn't a lot of, it doesn't sort of hold that mark. Phosphorus, again, we don't quite see the same decline that we saw with the wildfire, but you do see that self-thinning increase that happens from around 40 to 60 years. And then if we move to the salvage largely follows the wildfire. You'd probably expect that, you know, it's certainly dominated by that, but we do generally for all of these elements, we do see that there is a greater decline that happens. And like, so carbon, it's maybe an extra 10 tons per hectare that we're losing. We see a bit more nitrogen, and the same thing with phosphorus, that decline that we're seeing out to, 30 or 40 years when we looked at the wildfire, we see certainly out to 20 years that it's definitely lower. Oh, now I'm gonna have to go back because it's such a critical slide. No, there we are. I just, in keeping with our theme of linking above ground and below ground, I just wanted to show that the stand development in terms of, in this case, it's a gross total volume, so stem volume over time. And again, the fire is the green, and the blue is the harvest, and generally they pretty much are tracking right on top of each other. But certainly, if we look at the salvage at this point in time, it certainly is lagging behind, and that could be a combination of low stand density, maybe because of the burn and the removal, as well as some slower growth. So just to wrap up here, the take-home messages: certainly no evidence that operational clear-cut and generally full tree logging method, which is we use here in Ontario, results in any long-term declines in soil, carbon, or nutrient stocks. When we contrast the wildfire harvest with the double disturbance, the salvage logging, though, we do see those additional declines, and we see little evidence of recovery, at least out to 20 years. Again, we don't have long-term measurements on the salvage, and that's sort of our third point, is that we probably really could use some long-term monitoring on these salvage log sites to maybe better describe the fuller extent of the recovery patterns that we may expect to see, or whether or not they're going to continue to lag behind. And that's it, Deb.
We are going to talk about temperature changes and how that alters fungal community dynamics today. First, I want to acknowledge my collaborators here, Mark Bannock, who really co-led this project with me, and Dan Lindner, and Matt Smith. So Mark and Dan are both at the USDA Forest Service with me in Madison, Wisconsin, and Matt Smith is at the University of Florida in Gainesville. Okay, so this is kind of a different avenue, I guess. It's not the right word, but different track from where the rest of the talks, or a lot of the rest of the talks have been today, at least, and we're talking about decay fungi, right? So why do we care about wood decay fungi? Like, you go out in the woods, you see this something like this log that's covered in nice fruiting bodies. Some of them are slimy and moldy, and some of them are nice and up. But the log is full of decay fungi, right? Like, not just what we see on the log, but things we see that are in the log that we cannot see fruiting at that time. Over 70 petagrams of carbon is stored in wood worldwide. So just to give you a frame of reference, that is the mass roughly of, I think it's something like 760,000 United States aircraft carriers, giant, right, massive barge-like things. So it's a lot, right? The release of carbon and other nutrients depends on wood decay fungi. So they're incredibly important. They function as members of a diverse fungal community, and indeed, right, diverse forest communities. And understanding fungal community dynamics is really essential for predicting forest ecosystem function and ecosystem stability. And this becomes even more important in a changing world. Like, there's so much we do not know about fungi. And if we don't learn it now, we may not have the opportunity actually to see what communities are out there at this moment. So decay fungi are important for carbon cycling. Hopefully you got that a little from the last slide. But decay fungi are divided up into kind of a spectrum of classes, moving from brown rot fungi to white rot fungi. And that depends on, the classification depends on what they do to lignin and other nutrients in the wood too. But brown rot fungi really leave more lignin behind, while the white rot fungi leave less lignin behind. And in between brown and white rot, there's this other spectrum of rot types, right? You might hear soft rot or gray rot, et cetera. It's not, it's not kind of a white or brown world, right? There's a whole diversity, but we'll be talking about these two main types. All right, so brown rot fungi shuttle more carbon into the soil, and the white rot fungi return more carbon to the atmosphere. All right, so they're performing carbon cycling and returning the carbon in two different ways. So this led us to our main question here for this research. So our lab really focuses on decay fungi. We love wood decay fungi. And so we thought, okay, so how do changes in temperature affect fungal communities and ecological outcomes in decaying wood? And that question branches off into two kind of main research questions for this work. So first, do fine scale changes in temperature, things like 4 degrees C and 2 degrees C, matter for fungal community composition and development in wood? And do we also see kind of an effect of long-term carbon residues, how carbon cycling might be impacted by the decay fungi, or do we really just see this with short-term decay rates? So to do this, you see this picture of our research site. Kind of, this is a stand of very small aspen trees up in Herbster, Wisconsin, United States. Herbster is a small town that borders Lake Superior in northern Wisconsin. Okay, so up there, we collected small diameter aspen and balsam. And so you can see these little small diameter stems here. From these stems that were collected, we prune them off in the lab and cut these stems into 1 centimeter discs, okay, of the log. And we term these micro logs. So this is our sampling unit, right? So we collected these small trees from northern Wisconsin, cut them. We had sister logs and all this and all kinds of controls, but basically we have these logs and then soil collected from the site. We have aspen and balsam, and we put these micro logs in a sterile incubation chamber. The incubation chamber was sterile. However, the soil that you see here at the bottom of the incubation chamber and the micro log, these were non-sterile to allow us to observe fungal community development and see decay rates. So from here, we had 100 total micrologs per tree species, right? And then 20 of these micrologs per species were kind of control logs. So for us to get initial properties, what was the initial fungal community? What was the initial density and initial lignin content, things like that. And so our test micrologs were incubated at 8 different temperatures for 7 months. And so these temperatures were 4, 8, 12, 16, 18, 20, 22, 28, and 34 degrees C. And so we looked at mass loss, right? So compared sister microlog from the beginning to a test microlog at the end. Lignin content, for aspen only, I should say. Mass loss we did for both balsam and aspen, and then fungal community composition via high throughput amplicon sequencing for both aspen and balsam. So I'm going to walk you through this high throughput amplicon sequencing workflow quickly first before moving into the methods, or the results rather, because I'm not sure how many people are familiar with this. Okay, so with this, what you do is basically you take your microlog sample, right? You extract DNA, you put it in your sequencer here, and out you get your fungal community, kind of, right? Like that's the nice story. But there's all these steps in between, and I'm not going to go into these steps, so don't worry. Basically, this moves in from sampling, right? You do all these things. We did PCR with some fungal specific primers. You do bioinformatics because at the end you get millions and millions of sequences. You have to process these down into your biological interpretation, right? Like what you want. What is your fungal community composition? And for this too, we use controls so that we have a little bit more trust rather in our results. And these are called mock communities. Okay. Without these, you don't really know if what you're putting in, right, your input here, your sample of unknown community composition matches your output. So we have a sequencing control where you know what you're putting in, you know what you're getting out, and you can kind of parameterize your sequencing and bioinformatics. And so with those controls, you no longer see a difference between your input and your output, right? So when I tell you, like, oh, at the end of this, we got something like 2,000 operational taxonomic units or OTUs from these micrologs in the soil, you know, like, wait, how could that possibly be? And can I trust that? We do our best possible to have you trust that. And so an operational taxonomic unit, I just threw that word out there, or that term, that is, you can think of that roughly as a species level ID. Okay, so right, so we did, this is our pipeline. So, and I'm happy to answer questions about this if anybody has them. So for our results, since I just told you about how we did the fungal community composition, we're going to do that first. All right, so you're looking at a non-metric multi-dimensional scaling ordination. This is just kind of a way to view community composition in space. So each of these dots represents a microlog, okay? So balsam are the squares, aspen are the triangles. You see the temperatures and the ellipses. So each dot represents a microlog, and each microlog has multiple fungal species or OTUs. Those that are closer together have more similar communities, and those that are further apart have less similar communities, okay? So what you can see here right away is our control micrologs are quite different, and we see a pretty big shift, right, at different temperatures for both aspen and balsam in community composition. And if we break this up and just look at aspen and balsam separately without these controls, right, we still see these shifts, and it's actually a little bit more prominent. And interestingly, right, so we had some 2 degrees C intervals, 1618, 1820, and 2022. And the 16 to 18 degree shift here and here, right, that's where the arrows are, they have the most turnover in fungal community composition. We see this actually more in aspen. If we look at the decay rates, aspen is on top here in the circles. Aspen decomposed faster than balsam. And you see this peak at 28C, right? This rapid increase in rate here. And there is also an initial shift between 16 and 18C. So 16 and 18 is popping back up. Okay, lignin we measured only for aspen, I said before, because we didn't see a significant increase in decay rates for balsam. So for lignin, things seem more or less level, right? Until we hit the 16 to 18 degree shift again. And then we start to see a decrease in the total lignin content. And that continues, right, until 28 degrees C. So this 28 degrees C, this appears to be an optimum. And white rot fungi here might be more important than brown rot fungi at 28. You see less lignin being formed, right? Which means less carbon is being sequestered, and you probably have less organic matter getting into the soil. And then at between 28 and 34, you see this increase in total lignin. At 34 is relatively unrealistic given our sampling site in northern Wisconsin in the United States. But everything is more or less shut down at 34. It's relatively artificial. Lignin is not being broken down here, and you might see actually more soft rots taking over. And the fungal community composition was completely different at this temperature in both aspen and balsam too. There was a shift, not just in lignin content, but also in fungal community composition. Okay, so to wrap this up, our future directions, we would like to look more closely at smaller temperature increments. Some of our increments were relatively large, right? Like 6 degrees or 4 degree increments, but we'd like to look closer, tune finer down, perhaps into half-degree increments, particularly between, I said here, 22 to 30 C range, but actually 16 to 18 is probably where we should really start the fine-tuning. And we'd like to follow up to look at temperature effects on specific fungi too, and really start to get into how much carbon moves into the soil from dead wood. Okay.
Debbie mentioned that I've been working with Biochar with her. Like most forest entomologists, you took me kicking and screaming below ground. I don't know how you soil scientists do it. It's way too hard to sample insects down there. And what we're mostly interested in my lab is how various soil amendments, and for this talk, it's going to be biochar, impact insects' ability, if you will, to perform their normal ecosystem functions. So with the ecosystem functions, we're primarily talking about decomposition and nutrient cycling for this talk, but my lab is also moving into how some of these soil amendments may impact things like the use of root tissue by biocontrol agents of invasive noxious weeds, and pollination above ground. Oh, now I can't get the slides to move. What is going on? It is not advancing for me. Oh, there we go. We've taken multiple experimental approaches, and I'm just gonna present pretty much an overview of things. We do small container exposure trials to determine what impact or lack thereof biochar has on various organisms. The two figures here on the left of the screen are exposure trials where these two are for two typical forest insects here. Ips pini is a sclitid, a bark beetle that attacks pine. Formica obscuripes is a common ant that we find in our forests. And we did exposures, and I don't want to go through all of these, but we did exposures where there was a control, where there was no biochar exposure. We added a vial to it, so there was something else in the arena, a vial plus mesh over top of it. And then we added biochar, and this last column is biochar alone. The take-home message here is that if you start looking at it, the only way that biochar has an impact on the insects, which is consistent across multiple groups, is if they come in direct contact with it. So we think one of two things is happening. Either it is abrading, that waxy covering on the cuticle, and the insects are starting to desiccate or nanoparticles of the material as the insects walk across them, rub up against them, things like that, are breaking off and starting to clog the spiracles of the insects and causing them not to be able to breathe, suffocating them. So this is a very consistent drop off, but they have to be in contact with the material. And it's important for me to emphasize that this is dry material. If you go below ground, which is what these arenas were for, and below ground being what, 1.5 centimeters, you have soil alone, biochar alone. We mixed the two, and then we did a surface treatment of biochar. These two insects that are listed are biocontrol agents for spotted knapweed that are released here and I believe in Canada. And one of the things that I want you to notice right away is that with the soil in the mix, with it not being completely dry biochar, there are no significant differences amongst the treatments. And we get virtually, well, with this root feeder here, we get basically 100% survival throughout our study period. We do see some mortality with this other weevil, but it's very consistent and it doesn't seem to be related to the biochar at all. Moving out into the field, we've been conducting a lot of small, I'll call them plot studies, where we use cut bolts. This happens to be ponderosa pine, where we take the butt 2 sections of the tree, I think these were 1 meter sections, and that represents a paired replicate for us. Within those plots, we will surface treat one with biochar as if we had treated an entire plot with biochar, leave it out for a designated period of time, bring it back into the lab so that things like buprestid, serum will be able to be reared out without being parasitized too heavily. And then we monitor over a period of time. So going back to Ips pini, like I said, it's a common bark beetle here in Idaho. It's common throughout the pine forests of Canada as well. And we wanted to ensure that these bolts were attacked by this beetle. So we baited them with a pheromone lure. And the purpose of the study was to see if they would even utilize these biochar covered bolts. The attack here represents the number, the density of beetle attacks on the bolts. Emergence represents the number of beetles that came out, obviously. And if you cut to the chase, there were no differences on if we had biochar on the bolts or not. So they were willing to go through the biochar, get into the tree, utilize it, introduce the fungal species that they bring into the picture, and start their part of the decomposition process of the tree material. The other thing that we felt was important to see is that this emergence, which is also a density measurement, they're emerging at exactly the same rate or density regardless of the treatment. So once the bolts are on the ground, if they're treated and if they're attacked, then we don't see any differences at all in if the bolts have biochar on the surface or not. These beetles go right through it. Now, doing it in a different way, we did the exact same types of treatments, a meter section of tree paired based on the bottom 2 bolts. And we put nothing on those. We put no pheromones on, did not want to induce attack. We're just gonna wait and see what came in and developed, was willing to attack and develop. One of the first things that we found is that when we don't have biochar on the bolts, we get a higher diversity of insects willing to go into those, willing to utilize them and emerge. These don't look like great diversity, but when you look at what the organisms are, wood boring beetles like longhorn beetles and jewel beetles, some bark beetles, we have sericid wood wasps. Even though it's only 3 species in any one bolt compared to just under 2 species in any one bolt, the difference is significant and very consistent. This is a list of the common and abundant members of the insect communities that we reared. And what I want to point out more than anything is that if you go down this, the bark beetles, which are the subfamily Skeletinae, and the curculionids, the weevils, turpentine beetle hit in huge, or emerged in huge numbers on the control bolts compared to the surface treated bolts. Now, it was a limited study in that we only had, I think it was 10 pairs of bolts in each. But if you look at what was attacked, there was only a single bolt that had 6 individuals come out of it for the surface treated, as opposed to, or had 3 beetles emerge from the surface treated, I'm sorry. And there were 6 of the 10, so 60% of the control bolts were utilized with well over 300 beetles coming out of them. Ips pini, which like I said, is a common species, seemed to not really care which was which. If you come down to the longhorn beetles, the cerambicids, they don't really seem to care. And with the buprestids, there is a slight preference for the surface treated. That doesn't really surprise me. A lot of them will go into burned material, so that makes sense. Now the sericids, the wood wasps, actually prefer burned material. So we find those directly following wildfires a lot, and so it's unsurprising that they would be in the bolts that were treated with biochar. Looking slightly differently when we apply to the surface and look at small plot communities, what we did was looked at ant communities before and following soil treatments, biochar and or fertilizer treatments in the woods. And what we see is that when biochar is present, we have the lowest alone, we have the lowest change in ant community structure. We have the highest change in when we apply both biochar and fertilizer to those plots. But the only one that stays, in my opinion, really close to the controls is when we do the biochar alone after a harvest. This is a sampling of a list of the ant genera that we captured, and that was consistent across sites. Going really below ground now, and this is the stuff that Brad Card, who is on this presentation, is responsible for. He's a termite specialist at Oklahoma State. And we looked at burned and unburned sites. This was in the southeastern part of Idaho in the Targhee Caribou National Forest. And if you go across, you'll see that there are pine billets and aspen billets that are on the surface or buried. And the way Brad has done his comparisons is he looks at the total number of billets that were damaged, and there's a consistently higher number of billets that are utilized in burned areas, and it when you get into it, you get very distinct termite damage. These were staked down on the surface. There's some termites that are actually feeding, and this paper, I think, was just recently accepted at Southwestern Entomologist. More importantly, from my perspective, is that if you look at the mass loss that the termites and decay fungi cause on buried aspen billets. This is again the Targhee Caribou National Forest site, and the decay loss from termites plus, or the mass loss from termites plus the decay fungi, are these slash hashed columns as opposed to the gray columns. And there's a consistent increase, which you'd expect, with the termites adding to what is the mass loss that the fungi are causing. So this was at relatively high elevation, at least for termites. And there's this steady increase that I mentioned in mass loss due to both termite and decay activity, but the termites contribute somewhere between 20 and 48% to mass loss over if the fungi are there alone. So, wrapping it up, both fire, controlled or wildfire, and the use of biochar applications influence the use of woody material by wood inhabiting insects. Many of these insects provide ecosystem services in the way of nutrient cycling and beginning the process of wood decomposition that, as Michelle spoke about just before me, is carried on primarily by microbial agents, but without the insects, that the insects allow them to penetrate a little quicker into the wood. Consequently, the changed usage may impact what I'll call typical patterns of decomposition or nutrient recycling. If the insects are boring in and these microbes can follow them into their galleries, penetrate a little deeper, a little quicker, have more surface area to work on, we think that may be playing a large role and maybe changing things rather dramatically. These impacts can be seen as changes in host usage. Examples would be the termites on burn sites, some of the bark beetles that we've examined, overall adult survival. If you look at exposure to dry biochar, something I didn't talk about was a larval development study where we actually forced Douglas fir tussock moth, a common and serious defoliating pest, to consume small quantities of biochar with their, when being reared on synthetic diet, and it decreased survival dramatically, as well as weight gain. And then these changes also can impact community structure. So our future work is going to address the scale of these impacts and overall changes in the insect communities. And as I mentioned, we're looking a lot at how it impacts feeding by biocontrol agents, and we're looking at how it may impact plant health and have that reverberate into the pollinator community. And that was my last slide.
So good afternoon. I'm really pleased to be here on a Friday afternoon to try and distill some of the findings from several studies over the last 10 years that have been carried out by some pretty talented graduate and undergraduate students. And what we're going to talk about is this decade-long experiment that has been running to look at the effects of the byproduct, or really the waste product, of bioenergy production, which is wood ash, to answer the question, is it trash or is it treasure? So this experiment was established in 2012, just outside of Thunder Bay. And so I have a little map here showing where Thunder Bay is, if you're not familiar with where Thunder Bay is. It was started by Dr. Nancy Lukai and her grad student, Robin Sabine, who's now Robin Cooch. And as an aside, this was actually the year that I started at Lakehead. And so there's 9 experimental treatments where ashes from power boilers 3 and 6 produced from Resolute's Thunder Bay operation were added to the soil at a rate of 1,000 kilograms per hectare or 10,000 kilograms per hectare or none, both alone and in combination. And so you're going to see them being referred to as low and high carbon wood ashes. So plots, each plot was split in half. Half was planted with black spruce, half was planted with white spruce, and each treatment was replicated 5 times. So the objectives were, and they continue to be, what is the effect on soil properties and the growth and foliar nutrition of these commercially important species. So starting out, Robin measured the height of the seedlings. She collected foliage and measured 100 needle weights and contents of total nitrogen, phosphorus, calcium, potassium, and so on in the foliar tissue. She also measured soil bulk density, pH, electrical conductivity, exchangeable, base cations, basically, cation exchange capacity, total and extractable metals, microbial biomass, microbial respiration, mineralizable nitrogen. So a whole suite of parameters in the soil. And so when there was a significant ash treatment effect, we used orthogonal contrast to ask questions about the ash additions themselves, starting with, does the addition of ash have an effect? Does the effect of low carbon ash differ from high carbon ash? Does the amount of the low carbon ash matter? Does the amount of high carbon ash matter? And so on. And these contrasts are something that we've been carrying through with the various studies, and Dave has been really patient with me. So not surprisingly, the addition of ash increased soil pH and electrical conductivity. The low carbon ash is also the most reactive. It was produced at a higher temperature with a more complete combustion. It had a greater effect, and the effect was greater still when you added more of it. Also not surprising, when it was added to the high carbon ash, it enhanced the effect, again, because it is quite reactive. So these findings carry through to exchangeable calcium, and though the ash treatment didn't differ from the no ash treatment for exchangeable potassium and total sulfur, the low carbon ash had a greater effect than the high carbon ash, and generally the more that you add, the greater the effect. So with extractable iron and manganese, we can see the effect of the pH increase on the concentrations of these elements. So usually the addition of ash lowered the concentration of these elements, but we know that the solubility of these elements decreases with increasing pH. Trends in extractable zinc reflect the addition of the zinc from the ash itself. So the addition of ash resulted in a smaller microbial biomass carbon pool. So that's what we see in the top panel, but one that had a higher rate of respiration. And so that's what we see in the bottom panel. And the type of ash didn't affect the pool of the size of the pool, the microbial biomass carbon pool, but respiration was higher in the high carbon ash treatment. And even though the addition of more of the high carbon ash resulted in a smaller microbial biomass carbon pool, the rate of respiration wasn't affected. And when the high carbon ash was added to the low carbon ash, when that ash was applied at 1,000 kilograms per hectare, both the microbial biomass carbon pool and respiration increased. So this could suggest that the high carbon ash may actually be being used as a source of carbon by the microbial community. So we did see a lot of effects in the short term, not surprisingly, and the application of that low carbon ash, which is more reactive and at higher loading rates, had the greatest effects. High carbon ash also increased microbial respiration, and it may be acting as a carbon source. So my own research, or my research program, focuses on soil organic matter and specifically soil organic carbon. And I use a variety of indicators in my work to try and untangle the effects of management and environmental change on the soil organic carbon pool. So this dovetails quite nicely with investigating soil quality and soil health. And in 2015, where, well, AshNet was already established, and there's been several presentations this week about AshNet, which is that network of ash addition studies that span Canada that Paul Hazlitt led. And in 2015, as part of a graduate course, two of my students and myself used multiple indicators to look at the effect of ash addition on the soil. And this was really meant to be a case study for, whether we would look at it across sites. So 3 years after ash addition, we found mostly no effects on the soil properties that we measured. So no effects on things like carbon and nitrogen storage. The pH effects were largely dissipated by that point, not seeing any effects on microbial biomass, carbon, or respiration after 3 years. We did, though, find that the application of that high carbon ash did result in significantly higher carbon concentrations in the whole soil and also in the sand size fraction. So that's meant to be that uncomplexed fraction of organic matter, more coarser textured. And so we also saw wider carbon to nitrogen ratios in both the whole soil and the sand size fraction, again, consistent with the incorporation of that material that has a really, really wide carbon to nitrogen ratio. We saw wider ratios of respiration to microbial biomass that might be suggesting more efficient decomposition of organic matter, and significantly lower ratios of hot water extractable to total carbon and microbial biomass carbon to total carbon. But those ratios are quite complementary, so it wasn't entirely unexpected. So generally, no effect of ash addition on our soil health indicators. There were effects for the high carbon ash, but only at that high rate of application. So that's suggesting that carbon's retained on site and in that sand fraction. There's a lower proportion of carbon in the microbial biomass carbon pool, but the community may be working more efficiently. So you saw Dave earlier today, who gave a really great talk, and Marty and Alyssa are also in this figure. So Dave and his team have been measuring the heights and stem diameters over the years. And what we've seen looking at that data is no effect of ash addition on the height and volume of white spruce, but positive effects for black spruce. And those effects tend to decline through time. And really by 2017, so 5 years later, those effects were really starting to dissipate. So we submitted a successful application to the Canadian Wood Fiber Centre's FIP call, and that was to reapply ash. And this involved first doing some baseline soil sampling. So 6 years post application, the carbon to nitrogen ratio was still wider in the soils that received ash, but it was really being driven by that high carbon ash, and the more you apply, the greater the effect. And though there was no significant effect of the ash application on pH and electric conductivity by that point, there were significant effects for some exchangeable and extractable elements. And so you can see here, this is exchangeable calcium, and I just have up in the top is that initial figure that I showed earlier. So you can see that calcium concentrations have declined, but they're still higher in the low carbon and treatments where loading rates were higher. And same for extractable zinc. There were also significant differences for extractable manganese, exchangeable potassium, and total sulfur. Again, all 6 years after application. So not surprising, many of these effects actually carried over, and we see them again with the reapplication of ash. Work is still ongoing, and we've been continuing some of those remeasurements. I mean, the pandemic, of course, kind of buggered that up a little bit. So I've had a master's student working on this project, and just as I kind of, I'm wrapping things up, he did see significantly higher concentrations of carbon in the soil in the year after application, and that still followed through into 2020. So potentially seeing increases in carbon concentrations in the soil with those large ash additions. So what did we learn? Well, ash chemistry certainly matters in terms of the effect that it's going to have on the soil. And not surprisingly, the effects are going to be species specific. And though we don't see all chemical changes persisting over many years, some do. And that is important depending on the goal of the application. And overall, we see really no detrimental effects to the soil or the trees, which is a win in my books. So I would view it as treasure, not trash. And we're just going to keep monitoring. So just want to thank all the funders that are involved in this project, and in particular, Dave's crew as well for helping to keep this running. So thank you very much.
Basically, this title is pretty long, and I wanted to boil it down to basically fungi above and below ground, because that's what I'm going to be talking about, and how they respond to forest management practices. This is some work for my PhD, and of course, if anyone has worked with a PhD student, they know that it's a labor of love, and it takes a whole village to raise a student. So thank you to all these folks, and I'll highlight a few as we go along. I'm going to talk a little bit about why we were doing this research, and then talk about how we look at forest fungi above ground and below ground, and what that means, some of the comparisons, implications, and future directions. When we're looking above ground fungi, we're looking at all fruiting bodies, things that you would expect, but sometimes fungi that you wouldn't expect are fungi. DNA sequencing, finding the fungi that are within these wood stakes, and then the comparisons. So a little background, Western United States forest management involved fire suppression, which led to overstocked forests. So some of the biomass that we have in those overstocked forests can be feedstock specifically for things like bio-oil and biochar, as Debbie has talked a little bit about. The picture on the top is the biochar. It's kind of dusty. It can be small or large in particle size. And the bottom image is one I took of the biochar under scanning electron microscopy. And you'll notice that the shape is very jagged, and we'll get back to some of the imagery of what that looks like. Here's a blown up version of that biochar, which is usually very recalcitrant to decay. This is another good time to bring up my thanks for working on this project with all these people who have brought in their expertise from many different areas and avenues, including my advisor, Dan Lindner. So the reason we're doing it is because there's policies for use of the biomass for bioenergy. We have guidelines to maintain productivity in sites that involve tree species and soils. And they also include the retention of dead woody materials, because those materials create the base for the next forest. Decay fungi are modifying the wood in the same way that Steve was talking about insects modifying the wood. They're breaking it down. They're recycling the carbon and creating soil humus, and that dead wood is the base of the food chain. Fungi are very important for this mutualism that allows plants especially to get going. All land terrestrial plants got going with mycorrhizal partners because they helped the plant get water and nutrients. But sometimes fungi can be complicated. So here's a picture of Amphimina bisoides. It's a mycorrhizal fungi that we found on charred wood, but it's also a white rot fungi, and that's how they obtain their nutrients as well. Which leads into fungi ecosystems as pathogens. They can parasitize and kill their hosts and drive forest succession. The top example is Heterobasidian root conchs, which cause a root disease and a butt rot. and Phylum Ascomycota in the bottom, which causes a laminated butt rot. This is the brown rot that's caused by Phylum Ascomycota, and you can see that crumbly look is very characteristic of brown rot, as Michelle described when the white cellulose is missing. This is a very United States centric map, but it shows you where we are in Idaho. This is the Nez Perce area, and our sites are dominated by pine and fir species. I worked with biomass treatments that involve the thinning and removal of biomass and or the addition of the biomass to look and see if adding twice the amount of biomass is going to have any effect on anything, basically. So you'll see the difference in thin versus non-thin trees. We reduced them by about a third. These plots also had soil amendments in the form of fertilizer, biochar, and then a non-treated control. We did this on 400 meters square plots in 16 different treatment combinations. So above ground fruiting bodies, we looked at everything. Anything that was on the ground that was woody, we sampled for fungi, and we looked at what the fungi was on, how big that piece of wood was, and how decayed it was. And then if we didn't identify it in the field, we took it back to the lab for identification, which means I did a lot of microscopy. What I used to think was white bird poop on trees or wood was actually something. And so I got really talented at this thin sectioning microscopy. And once you get under the light microscope, you can see these huge differences. This fruiting body is over 100 microns wide, and that's how decay gets started, because these crusts start to form. But it meant I did a lot of microscopy to try and identify some of the differences between what's there and what's not. To analyze this data, we had a little difference because normal diversity indices are just not appropriate for fungi. For example, Armillaria, the humongous fungus, may take up hectares. And is it a discrete organism? Maybe not. So the assumptions of normality that we have with fungi are different than with, say, birds or humans. So I'll talk about those tests, but really we did non-parametric tests, as Michelle had already explained. What did we find? We found a lot of pathogens. We found things that came right away, like Cryptoporus vulvatus, that white puffy fungi, and it's associated with a lot of insects that like to come and spread its spores, but it's only found in those early stages of decay. And then as you see in forests, there's this succession of fungi that move through as the wood becomes more decayed. These are species accumulation curves, and they just show that we didn't find all the species that we could because the curves never leveled out. We had a significantly wet year in 2016, but we could definitely find more fungi if we sampled more. There was a little difference between the sites, but if you look on the axis, you'll notice that the mean species richness difference was only a few species with a lot of overlap. There was some treatment effects the first year, and it seems to suggest that by removing the biomass, you might see more fungi. But these graphs mostly indicate how much variability there are with fungal fruiting body data. We didn't see a significant difference with the soil amendments with fruiting bodies, and there was great variation between the treatments. We did observe over 1,000 observations and over 100 different species, and a lot of them were basidiomycetes, which we have a lot of information about. By the third year, we observed more mycorrhizal species, like Amphimina, which you see here on the right. And biomass treatment effects were significant the first year, which makes sense because there was a huge logging event that happened. And we were able to detect important tree pathogens. We also provided collections and sequence data for rarely studied fungi. But there's non-normal data because what size logs were originally on a plot weren't manipulated. So if there was a big log on one plot and not a big log on the other, that's just the way it was. The second part of our study was to look at the fungi that were in the wood stakes on these same plots. And the wood stakes were on the surface, stapled, or located within the mineral soil 30 centimeters down. So we have this divided into a mineral top and a mineral bottom layer. Basically, we would remove the stakes, we would drill them with a drill, and then we would store each bit of wood shavings for DNA extraction. I used the same high fruit amplicon sequencing methods that Michelle did, so I'm really happy that she went through and described all these for us. And what did we find? We found that most of our abundant OTUs via DNA analysis were from this Phylum Ascomycota, which means they are ascospores that are formed in an asci, and they reproduce asexually. We don't know that much about them. We know a little bit about their family, class, and order, but you'll notice that we don't know a lot about them to the genus and species level specifically. And a lot of the ecology is unassigned, or we think it's saprotrophic, but it could be pathotrophic or symbiotrophic. So that explains the degree of awareness we have about these. But the analysis via non-parametric permutational ANOVA comes out quite cleanly. You can see there's a very distinct difference between our aspen and pine stakes. That's very clean. You can see a very distinct difference between the communities that are at the surface of the wood stakes versus the top and the bottom of the mineral state. The surface community is on the far right, that dark bluish community. And there's a lot of overlap between the fungal communities that are located in the mineral top and bottom. There's a lot of overlap between the years, but statistically speaking, there was a statistically significant difference between the fungal communities by year. And if you look at the table at the surface level, you can see those differences are very significant with very significant p-values. So the overall conclusions are that there's over 2,000 OTUs that we found in the wood stake analysis, but a lot of them are from Ascomycete species. There are significant differences between the aspen and pine stakes, and there are significant differences by stake location. The biomass treatment effects were significant all 3 years, and the soil treatment effects were significant all 3 years in the aspen surface stakes. What does this mean? What's the differences? Well, if we look at it, we had a lot more species that we identified via DNA analyses than the fruiting body surveys. Again, those are OTU comparisons, but there's a big difference. And what does that mean? Because there's more OTUs, there's a lot more statistical strength to talk about differences. So there was a significant difference every year in all the treatments that we found with the DNA analysis compared with the fruiting body analysis. We know a bit about what the fungi are and what they're doing in the fruiting bodies because of years of research. So we know that, for example, Trichaptum aviatinum is a white rot fungi. And when we look at some of the OTUs of the DNA analyses, we know less and less about what those fungi are doing because we haven't had dedicated mycologists that have spent as much time because they're so small and they're quite hard to work with. So a lot of the species are different that you'd find in the fruiting bodies versus the stakes. And even the Basidiomycete species, there's very little overlap of the species that you'd find because the substrates are so different. The wood stakes are just a very specific type of substrate. This is a Venn diagram showing the differences in sequences, and only 1% of the OTUs had overlapped. And Asterium sanguineolentum was the only forest pathogen that we had detected in drilled pine stake sequences. Again, that's a red streak rot fungi that comes in immediately after any sort of injury, so it comes in right away after logging. So the conclusions are fruiting body surveys, they give you fewer species than DNA analyses, and the data tends to be very non-normal. But we know quite a bit about the fungi, including the pathogenicity of it and what it's going to do, or in some cases, what some fungi are going to do. And DNA-based analyses are very sensitive to treatment effects. And so the utility of each method and what you want to do or sample for depends on the questions being asked and the skills and definitely the budget of the research team. So forest management objectives with fungi may involve IDing diseases of sight and then planting resistant species. For example, heterobasidian species favor resistant pines and larch that move in because the infected species are infected in their root systems. The same thing happens with Phylinus. Phylinus is very virulent to fir and hemlock trees. So therefore the next successional forest will involve resistant pines and larch species. So being an informed manager, you would plan for that. Armillaria ostoyoi is another one we found. It's the humongous fungus and it invades living root tissue. And it's been found with an increased incidence with fertilization because nitrogen is decomposition's limiting nutrient. And this is an example of armillaria on the landscape scale. So fungal DNA-based studies are very sensitive and responsive. So when people are asking, did my treatment do anything? Did adding ash do anything? Using fungi can be very informative and responsive because you may be able to see very slight community changes due to organic matter and tree species, disturbance, and pH. Oh, and the implications are we found many new species. So are you interested in naming and describing new species? This is Karen Nakasone. She's amazing. She's probably the last person that knows crust fungi, and we need a lot of help in that avenue. And can we relate communities to actual functioning in the environment? So the longer, broader term studies, just like Debbie's study, is what happens? What happens with these brown rotted woods? What happens to long-term productivity or stabilization? And how does wood become soil? This is our picture of char. This is our brown rotted wood. This is our white rotter on wood. And what does that look like in the future? So do you have land, soil, tree, biota data? And are you interested in looking at some of these relationships with fungi? There's almost guaranteed significance. Are you interested in insects? This is for you, Steve. These are some pictures that our SEM team took of termite parts. And we just watered for our termite butt cones there. When do you want to sample? You're going to want to sample when you have enough moisture in the area. And any climate diagram can tell you the perfect time. Again, thank you to everyone that helped on this. Thank you.